there are a lot to talk about here. It's a really exciting field. There are 3 sort of major manufacturers of this and many others that are in the wings. You know, Fred, you're involved with the KITE trials, the ZUMA1 study. You know, how's that going? I know there's a phase 1 that's been completed and now into the phase 2.
Yeah, it's going very well. So the ZUMA1 study is sponsored by KITE Pharmaceuticals. They've licensed the same construct that Jim Kokendorfer and Steve Rosenberg developed at the NCI. They provide centralized manufacturing for those CAR T cells. So we collect the T cells by apheresis, ship them out fresh to a centralized manufacturing facility where the CAR T cells are manufactured, frozen, and shipped back. The results of the phase 1 portion of that ZUMA1 study have been released, and essentially 7 patients were treated, and that was enough to determine that the fludarabine and Cytoxan dosing and the number of CAR T cells administered were safe. All patients who had T cells apheresed had successful manufacture of their CAR T cells, so that's important. And the turnaround time for those CAR T cells was relatively short at just over 2 weeks. So we're getting the cells back relatively quickly. Out of those 7 patients treated on the phase 1 portion of the ZUMA1 study, there was 1 dose-limiting toxicity. However, that patient died of an intracranial hemorrhage, and it was deemed by the investigators to not be due to the CAR T cells, but related to the lymphodepleting chemotherapy, and also how quite ill this patient was. Also had a sepsis at the same time when they passed away. The other patients, all the toxicities were reversible, including both cytokine release syndrome and neurologic toxicity. And all patients, 100%, did develop some neurologic toxicity. There seems to be sort of a dual peak in the neurologic toxicity, an early neurologic toxicity which occurs within 1 to 3 days after infusion of the CAR T cells, which is probably related to the cytokine release syndrome occurring at the same time, which is high fevers and chills. And then a later neurologic toxicity 7 days around that time, which is the sort of well-described locked-in syndrome or aphasia that's seen with all the different CAR T cell constructs. And again, these neurologic toxicities were reversible. As far as efficacy, it's only 7 patients, but out of those 7 patients, there were 4 CRs, actually 5 responders and 4 CRs, and 3 of those patients have durable CRs out to 9 months and counting. So, you know, what's really important about understanding this trial, so if I didn't mention it, it's for patients with diffuse large B-cell lymphoma, as well as primary mediastinal and transformed follicular lymphoma. But what's really important about this trial is that only patients with truly chemorefractory disease were enrolled. And as presented here at ASCO recently, the SCHOLAR-1 study, really, we all know as clinicians that a patient who doesn't respond to their last chemotherapy, a large cell lymphoma patient who doesn't respond to their last chemotherapy, has a very poor prognosis. But by collecting data from 4 different large databases or prospective clinical trials, the SCHOLAR-1 study showed that the overall response rate you can expect from a patient who didn't respond to their last chemotherapy or who relapsed within 12 months of an autologous hematopoietic stem cell transplant, you can expect an overall response rate at best 28–30% and a CR rate of like 8 to 10%. So here we have a trial using that same criteria to enroll large cell lymphoma patients, and 3 out of 7 had CRs. So it's pretty remarkable, and the phase 2 portion is ongoing and enrolling.
So I think that one of the other impressive things about that study and the other studies that are going on is not just the clinical results, which are fantastic, but also just the logistical advancement that we've had in terms of taking these out of single center academic and being able to do it in large trials across the United States. Now, Krishna, you're involved in the Juno trials. And,
That's right.
And what's your experience there?
Yeah, so we've been involved in both the KITE and the Juno study. The Juno rocket study is looking at the same principle, basically a CD19-directed CAR in the setting of ALL. There are a few differences in terms of the design. Actually, the antibody recognition fragment is actually the exact same as in the KITE study. There is also a CD28 co-stimulatory domain in the JCAR015 product, again, which is the focus of the first ALL study. The Juno B-cell lymphoma study actually uses a 4-1BB domain. Juno has decided that they would pre-select the cells. So with the JCAR015 product that's in the Rocket study, there is CD3 selection as opposed to an unselected product following the expansion of those cells. In the lymphoma study that Juno will be doing, actually, they're going to select both CD4 and CD8 cells as opposed to just doing total CD3, which is a generalized marker of CD3 cells. There are some other differences in the Juno Rocket study. There are 2 planned infusions of the CAR T cells. So you have basically a dealer's choice chemotherapy or supportive care once the patient is enrolled during the time that the manufacturing happens, and then you have lymphodepleting chemotherapy, which again right now the backbone is basically fludarabine and cyclophosphamide for most patients, and then an infusion of the cells, and again assuming that the patient has acceptable performance status and toxicity with the first cellular infusion, there's a dose escalated second infusion. So that is a little bit different than the design of the KITE study. So there are 2 infusions that are dose escalated.